Hi, I'm Chloe Imbrasard, the representative of the HKUST iGEM team of 2021. Today, I will be talking about biotechnology as our future, the iGEM competition. So actually, who are we? We are the current team members of the iGEM team of the Hong Kong University of Science and Technology. So what is iGEM? We know it is a competition, but what is it really? iGEM stands for the International Genetically Engineered Machine Competition. It is the first international synthetic biology competition for university undergraduate students. It lasts 1 year and teams have from 5 to 20 plus members. However, lots of high school teams in Hong Kong are also joining. So what do we do in the iGEM competition? We engineer new genetic parts, try to create something beneficial for the future, collaborate with other teams, and publish our findings at the end of the year. We find the problem we see in our local community and try to solve it using synthetic biology. Because we are doing this all around the world, we end up solving local problems everywhere. To show how big this competition is, this year there are 364 iGEM teams around the globe with 46 countries participating. There are also 13 different tracks or themes of projects for all the teams to choose from. These different tracks range from environment to therapeutics to manufacturing and even hardware. So there's definitely a theme that anyone can be interested in. To give you guys a bit of an idea about what iGEM project would look like, here there are some past team projects. First, there's a Team ETZ Zurich that created the coli sweeper, making a minesweeper game using E. coli. Team TU Kaiserslautern created Clammy Yummy. They engineered an organism with 2 plastic degrading enzymes. And Team TAS Taipei created Viral Spiral, a system for viral detection. Team TAS Taipei is actually a high school team. There are different modules or groups that can make up a good iGEM team. There is wet lab, where the experimentation and lab work is done. Circuit design, where we design the genes. Dry lab, where we do modeling, maths, and calculations. Hardware, for creating a tool, machinery, or other equipment like a detection kit. Anything that includes physical engineering falls into this category. There's Wiki, for the coding and creating or designing of the web page, and IHP, or Integrated Human Practices, which includes communication, education, and collaboration with other teams and other marketing aspects. So now, let's move on to our own project at HKUST. We are called Shelby this year. What are we making? A prokaryotic biosensor for detection of paralytic shellfish toxins. It sounds so complicated, right? So what is it actually? Here are some definitions of the words you saw previously. Prokaryotic or prokaryote. That is an organism that is composed of one cell that does not have a membrane covering the nucleus. Biosensor. This is a device which uses a living thing or a biological system to detect the presence of chemicals. Paralytic shellfish toxin, or PST. These are the chemicals that we are trying to test for. So, who likes seafood? A lot of us in Hong Kong love eating seafood like oysters and lobsters. However, did you know that some sea animals like shellfish, crabs, and even fish have the ability to poison you with toxins? Shown on the screen is the chemical structure of a PST, but in reality, they are so small you cannot even see it. As shellfish is a colloquial term for many types of marine animals, here we define shellfish as bivalve mollusks, and most of them are filter feeders. As they consume little phytoplankton and zooplankton, they also take up organisms that can produce life-threatening toxins. Now, these toxins are not toxic for the shellfish itself, but for the animals that consume it. Many shellfish can accumulate toxins as waste or even as a defense mechanism for up to 2 years. You must be wondering, where did these toxins come from? PSTs are actually created by small organisms found in harmful algal blooms. As you can see in these pictures, they usually cover large surface area and float on the top of oceans or lakes that have increase in nutrients. They are all very pretty in color as they could be green, red, or bioluminescent. These blooms, however, can suffocate fish and other animals, killing them and creating dead zones in fresh and salt water. HABs are also increasing around the world due to climate change and global warming, as well as increased water pollution. What do these toxins do to humans? PST blocks the sodium channels where cells intake salt of nerve cells. The symptoms of paralytic shellfish poisoning include headaches, vomiting, vertigo, all the way to paralysis, respiratory failure, and even death. We at Shelby are trying to engineer an organism to act as a sensor to detect paralytic shellfish toxins in shellfish. The basic mechanism is that if PST is present in shellfish, our sensor will turn red, and if PST is absent, it will turn green. In the presence of XTX and VTD, the sodium channels will not have a significant difference, which will trigger the expression of the genes responsible for the red fluorescence in E. coli. In the absence of STX but the presence of VTD, there will be an influx of sodium, which will trigger the expression of genes responsible for green fluorescence. As you can see, the possibilities for iGEM really are endless. Please contact us. We would really love to answer any questions or read any comments you have on our project or iGEM as a whole. So feel free to reach out to us through our email or Instagram. Thank you.